right. Hello, hello. Welcome, everyone. Welcome. And it's wonderful to see so many people here interested in such an amazing topic that at Northwestern is, leading on so much of this amazing work. I'm Kristen McDonnell, and I'm on the Northwestern University Board of Trustees, and I'm the president-elect of the Northwestern Alumni Association. And I would love to see how many alums do we have out in the audience tonight. Woo! All right, go Cats. Excellent. It's wonderful to see so many of you here. I studied industrial engineering and computer science in the McCormick School of Engineering at Northwestern in the class of 1985. So this topic is somewhat foreign to me, but I'm very excited to hear all about it. I'm also the parent of 3 daughters who are classes of 21, 23, and 25 at Northwestern. And yes, keep that tuition bill coming, Northwestern. And I'm super excited tonight that my sister is here, Dr. Gail Oslison-Kang, and she's a neurologist, and she is Northwestern class of 1990 in Feinberg. And with her is her son, who's incoming freshman Northwestern class of 2027. So Gail Kang and Ryan Kang are here tonight. So as you can see, we're a big, happy Northwestern family. So for those of you who went to school on Northwestern's Evanston campus, how many were just on Evanston campus? Okay. Did you know that there were other campuses? Okay, all right. So it turns out, as you may know, there's this Chicago campus that all this amazing work is being done on. There's also a campus here in San Francisco, and also a campus in Qatar, and in the Middle East. So 4 Northwestern campuses. And that what's happening across all these campuses, and especially with our new president, Michael Schill, there is just all this amazing collaboration, cross-pollination, innovation happening across all these Northwestern campuses. And so really excited to hear about what's happening at the Chicago campus tonight. So one of the things that we're finding is that through these innovative programs, and cross-collaboration, we may start to hear, I'm not sure if you're going to talk about the engineering school and the medical school at all tonight, but I'm starting to hear rumblings of that kind of cross-pollination is really, taking shape, and there's so many other ways that the collaboration across these campuses happens. So what we're going to do this evening is we have this amazing, stellar, interdisciplinary group of faculty from the Feinberg School of Medicine. And they're all looking at the same issue of longevity, but in different ways. So we're going to hear from, first, Dr. Douglas E. Vaughan, and he's going to introduce our panelists. And for a bit of background about amazing Dr. Vaughan, he is the Irving S. Cutter Professor of Medicine at Feinberg. He's the chair of the Department of Medicine, and most important for this evening, he's the founding director of the Potocsnak Longevity Institute at Northwestern University's Feinberg School of Medicine. Clinically, Dr. Vaughan focused on patients with coronary artery disease, and his research focus has been on the PI1 protein found in DNA and its impact on cardiovascular disease and aging. And this is the amazing, you're already amazing, but this is the really cool thing that I was like, this is really cool. Roughly a decade ago, Dr. Vaughan discovered an extended family of old order Amish in Indiana and discovered a genetic variant that protects them against multiple aspects of biological aging. And so over time, Dr. Vaughan discovered that these Amish had very low levels of PI1, and using that information and collaborating with a Japanese company, Dr. Vaughan developed an experimental drug that can prevent or block aging, and that drug is currently in clinical trials. So I just think that's really fascinating. So please join me in giving Dr. Vaughan a huge Northwestern San Francisco campus welcome.
Thank you, Kristen. You ought to just stay up here and do this. You're doing a great job. I like the way you introduce people. And thank you for your kind comments. Thank you all for being here tonight. It's a thrill to be here and to have an opportunity to talk to you about what's going on at the Potocsnak Longevity Institute. And it feels like home here. I mean, Northwestern on the wall, and you've made us very comfortable. And hopefully we can entertain you for a few minutes. At regular intervals, our Dean, Eric Nielsen, convenes a large number of our faculty to talk about the future and to talk about priorities and to talk about themes that we think we should be working on. And in 2016, we had one of those meetings with the Dean, and what bubbled out of that meeting were two thematic areas of intentional development and investment that we wanted to get involved with. One was around artificial or augmented intelligence, no surprise, and we have an institute around that. And you should hear from them sometime. It's pretty exciting too. And the other one that bubbled up was the idea that we needed to get involved and think about how we can impact upon aging and healthy aging in our species in a more deliberate sort of way. So out of that ideation, eventually the Potocsnak Longevity Institute was created with a generous gift from the Potocsnak family last year. And so we have some fuel in our rocket now to launch us and to actually move us into this domain in a way that we weren't able to do before. We're at a very unique point in human history right now, here in 2023. You probably already knew that. There are a lot of different things that make this a pretty unique point in time, but the convergence of our ability to measure biological age precisely has never been like it is today. We can precisely tell you your biological age in a way that was never possible before. And you couple that with the idea that we understand the biology of aging in a way that's never been understood, and those two factors coming together right now make us pretty optimistic that we can do something about aging in the near term, in real life, and that can impact upon the life and the health span of our species. Anybody ever seen this ad? A lot of these were on billboards around Chicago for a few years. The first person to live to be 150 is alive today. Let me look around the audience here. I'm trying to see. There are a couple of recent grads I saw come in there, class of 21 or 20. You guys maybe have a chance, but I sort of doubt it. It's kind of preposterous. But the reality is a lot of people are thinking about it. A lot of money is being invested in aging right now in ways that are just staggering and breathtaking. Bezos has put billions into it. The Saudi government is putting billions into it. There's a lot of crypto money in aging, no surprise. But it's all betting on some pretty good ideas and the big idea that we can actually do something about the process. We live on this planet. Look familiar to you? It's where you live. You fly around that sun every 365 days. You're flying at, this orb is flying at 66,000 miles an hour right now. You don't even feel it, barely feel it. But the reality is, and this is one of the points that we'll make tonight, and I think you'll hear it from our speakers as well, is that your biological age is not the same as your chronological age, right? And those two factors can differ dramatically in one person's versus the next. So, and that's what gives us an insight into aging, that gives us optimism that we can actually impact on it with interventions in a variety of different ways. So here's the slide, and now look at this. This is an important concept because this is a fundamental component of what makes us think we can do something about aging. So these are the trajectories of three different groups of people. The one in the middle are the average human being. They live their life, and their chronology pretty much matches up with their biology, and they're right in the middle there. And you might be on that track, I don't know. You might not. But on the other hand, we have these outliers in either direction. We have individuals that are super agers, people that don't seem to ever age at all. I'm sure you have some in your family, or you have some friends, or you have colleagues. You think, my God, they don't age at all. We had an event last night. One of our alumni, who's a rheumatologist here in town, came and he said, I'm going to my 70th Feinberg reunion this fall. I said, what? I said, you must miscount it. He said, no, 94. I still attend at UCSF. I still teach residents and fellows. And I said, what do we think he was? 70, looking at him? Angelo, what do you think? Anyway, pretty amazing, and totally with it. So he would go on that super ager category. The reality is, in the hospital, if you come into Northwestern and I walk you around the wards, I can show you a lot of people that have accelerated aging. That's the reality of medicine. And they come in all different kinds of flavors and backgrounds that drive that accelerated aging diathesis. I mean, the most common situation, clinical situation that drives accelerated aging is, I think, chronic kidney disease. If you take a 25-year-old on dialysis, they have the same cardiovascular risk as a 75-year-old, and I don't think we've figured out how to do anything about it. We see other common conditions that impact upon aging, chronic HIV infections, and that's part of what was the motivation for creating the Potocsnak Longevity Institute, and you'll hear about that in a few minutes. People that smoke cigarettes, people that have been exposed to toxic chemotherapy sometimes age more rapidly. There are other people with genetic syndromes that age more rapidly. So anyway, it's not all the same. And the fact that there are superagers makes us think that it's a malleable process, that there are things that we can potentially do to intervene to slow down the pace of aging in any given individual. So we're not, it's not like we haven't been studying aging and been interested in it at Northwestern for many years. It crosses many different disciplines, many different departments on both campuses in real ways. And you can see our portfolio from the National Institute of Aging has grown rather dramatically over the last several years. We're up to about 44 million last year total in funding from the NIA. That's not all the aging-related research that we do, but it's a good index of where we stack up in the world and how much we've grown and what our opportunity is. And we want to be one of the top 5 places in the world for doing this in a few years. And this gift and this institute, I think, is going to propel us in that direction. So 2 big important points on this slide. You don't have to look at all the different words that are on here, but the major hypothesis in geroscience is that aging is actually malleable, and that what we can do, what we hope to do by slowing the process of aging is deliver more disease-free years to people. We want to extend the health span, not necessarily the lifespan, but every disease that we take care of in adult medicine, every disease, the most important risk factor is age. Age is the most important risk factor for heart disease. Age is the most important risk factor for cancer. Age is the most important risk factor for cognitive decline. Age is the most important risk factor for pulmonary disease. You catch the trend? Okay, so if we can do a little bit about aging, we can push aging-related disease back a little bit and give people a little bit longer, happier, healthier lives. That's why we're doing this. This is a schematic representation of the Potocsnak Longevity Institute. As we ideated and as we created it, we came up with the idea that it would be comprised of several different centers under this one big umbrella. And I just want to mention them briefly. First, we'll have a Center for Basic and Translational Biology. We already do a lot of that on campus, but we hope we can catalyze and coordinate and synergize some of that work to grow it. We have a Center for HIV and Aging. That was, as I said, that was one of the catalytic ideas that motivated the Potocsnak family to give that big gift. We have a Center for Population Science and Aging. You're going to hear about that a little bit tonight as well. With McCormick and with Evanston, we have a Center for Nanoscience and Aging. You can't have a center at Northwestern without nanoscience. I don't know exactly what that means. We'll figure it out. But it might be wearables. It might be drug delivery systems. It might be ways that we measure biological age in ways that we've never done before. So we're going to bring that to the table. The one that's most interactive with the world at large, that interfaces with the public and potentially with you, is our Human Longevity Laboratory. So this is my idea to create a single facility where people can come in and have their biological age measured. So we have it all set up. We've actually started bringing people through here in the last week. What you do, you come in and we measure your biological age in a variety of different domains and different systems. It takes about 2 hours. It's nothing painful. Everything from your grip strength to your hearing to a picture of your retina to your pulse wave velocity to your heart rate variability to some molecular measures of your biological age, including your epigenetic age. And we do that all in a package. We give you a little report card and tell you where you are on one of those curves that I showed you earlier in the discussion. What do we want to do with that? Well, we're not just doing it for fun, although it's very interesting. We want to build a large cross-sectional database in the first place, and then we want to start doing interventional trials. Once we have that information, once we get really good at it, I think we'll have the most multidisciplinary, multifactorial measure of human aging in the world. And we want to bring people in and then do interventional trials. It might be drugs, it might be diet, it might be exercise, it might be combinations thereof. And we want to see if we can't change the slope or the velocity of aging in people that come in, particularly people that are aging more rapidly. I'm not too interested in making a billionaire live to be 150. That doesn't float my boat too much. But if I can help the average Chicago citizen in a way that impacts on their lives and their health and their health span, then that excites me a lot. And I think it excites our team a lot too. So the Human Longevity Laboratory is up and running. My big dream is, maybe one of you will be interested in supporting this idea. I want to build a network of these human longevity laboratories around the globe. Have one in every continent. And they do the same protocol, collect the same information, and do the same studies with us. And then we take the leadership role in the world in impacting upon human aging. We're already talking to a major hospital in Bangkok about this, and in Rio. We're picking good places. I have a little vote in that. But I hope that can really make Northwestern the leader in the world in this space, and it's certainly a lot of fun to be a part of it. So, the Fountain of Youth. We've been looking for it for a long time. Where was it? Where is it? It's not in Florida. I'll tell you that for sure. You know what? It's not completely in focus yet, but we're getting close. I think we can sort of see it. We can sort of see what its outlines. I think we're going to find it relatively soon. It may take a variety of forms. It may take a variety of different interventions to deliver that for people, but it's an unbelievable time in the history of our species, I think, to actually think about this in a credible, non-fantasy sort of way. So now, we're going to go into the important part of the program and listen to the speakers that we have with us tonight. We have 3 luminaries from the school to talk to you about their work and why they're interested in aging, how they hope they can impact on it. And they're also going to answer the question, is aging inevitable or is it preventable? And they've all got different opinions about that. So I'll bring them up one at a time and have them talk about their work. You guys can come up forward. Frank Palella is our first speaker. And Frank is a professor of medicine. He's the Potocsnak family professor of medicine as well. He's in infectious disease, and he's going to share with you his perspective on why he's interested in aging and what he can do about it. Are you going to stand here, or are you going to sit over?
We're going to transfer.
Do me a favor and scoot in just a hair.
Scoot in.
Questions after everybody finishes their little spiels, okay? So, Frank, you're on.
Thank you,
I'll sit here
Doug.
with you so you're
Thank you. I don't feel
comfortable.
lonely. Yeah. All right, Don.
Yeah.
You can,
Keep company, Frank.
you'll keep me company. Thank you all for coming tonight. It's hard to follow Doug, who is really
Oh, by the way, he's my co-director of the Institute. He's really, his relationship with the Potocsnak family really catalyzed this gift, and we owe him a great debt of gratitude for that.
Well, the Potocsnaks. I came to aging, the study of aging, the interest in aging, through treatment of persons with HIV. That sounds kind of funny, doesn't it? We used to think of HIV as being universally fatal. In fact, a mere 30 years ago, between 25 and 30, when I was much earlier in my career, AIDS was the leading cause of death among men in this country. It was third or fourth among women. It was horrible. We had young people dying every week, and little that we could do in terms of treatment. In the mid to late 90s, it became apparent that we could put together highly potent treatments that would keep people from dying, keep them from getting immunodeficient, suppress the virus, and the consequence was that we extended lifespan and survival by decades. In fact, it became clear with the most modern therapies, and I'm fast forwarding, single tablet regimen, one pill once a day, two or three or four drugs in one pill, that we could take people with HIV and allow them to extend their survival so that they came within 6 to 10 years of similar persons without HIV. But there's a catch, a big catch, is that persons with HIV tend to become stricken, even with well-controlled virally suppressed HIV, tend to be affected by the aging-related conditions like cardiovascular disease, liver disease, non-AIDS cancers, neurocognitive, dementia, about a decade to a decade and a half at an earlier age than people without HIV. Why is this? This has been the focus of my career and of many of my esteemed colleagues across the country, including a couple that I work closely with at UCSF and Stanford and other places on the West Coast. It's clear that even people with well-controlled HIV who are otherwise healthy have an excess of key processes that determine aging in all of us. It includes inflammation, blood vessel aging, and adverse effects on the immune system, even though the virus is controlled. So it became clear that HIV is a wonderful paradigm for accelerated aging. People with HIV, even when they're diagnosed early in the infection, and there's no such thing as diagnosing too early or starting on therapy too promptly, that the lifespan is a bit shortened, but the health span is significantly shortened. And through so the study of HIV and aging clearly has become the study of aging in general at an accelerated pace, so that the insights that we develop about aging in the accelerated model of persons with HIV are clearly applicable to persons without HIV who are aging at what we might consider a standard trajectory. So the three arrows that Doug put up, the standard agers, the accelerated agers, and the super agers, the persons with HIV tend to be accelerated agers, even though they may not be technically immune deficient anymore. This has created an energy around the whole idea of what can we do about this. Medications like anti-inflammatory drugs. Doug knows a lot, and we are doing clinical trials with the National Institutes of Health, even at Northwestern, of classes of drugs that a layperson might consider, well, are these part of the fountain of youth solution, what we call senolytic, xenomorphic, or senostatic drugs. We're studying them. There's no magic bullet yet. I universally say to new patients who see me, if you take your medication daily, especially when they're younger, you're going to live longer than I am. That was unheard of 25 years ago. The Potocsnak family came to my attention about 22 years ago when a dear family member of theirs developed very late stage AIDS. It wasn't known that he was infected with HIV. We were able to help him. He's now thriving with a large family of his own and doing beautifully. It is paramount importance to them, and I think to the 1.2 million other people living with HIV in this country, over half of whom are over 50, and by 2030, by CDC's estimate, over 3/4 will be over the age of 50, half will be over the age of 60. It is paramount to those people and to the 30 to 40,000 that are going to become infected each year, if current trajectories hold true, that we do things to allow people living with HIV to not only live as long as persons without, but to live as well as fully by all the indices that you heard Doug talk about, and you're going to hear our other panelists talk about. Functional status, the ability to think, the ability to thrive, the ability to work, the ability to be free of the cancers and the aging-associated conditions, which today are the principal determinants of how long people with HIV live. As you've probably guessed, I could go on and on. I'm going to limit my comments here, but other to say that the complementarity between the study of aging in general and the study of HIV among aging persons with it allows us to gain insights as to processes and as to potential ways to decelerate aging and extend health span. I'm going to stop there. And
You have to answer the question, though.
Okay.
Is it inevitable or preventable?
I think at this point, it's delayable. It's delayable. We're all born with genes that sort of set a genetic clock, or we call an epigenetic clock. And we're not going to get too microbiologically technical, although I'm sure many of you could speak the language.
Yes, we are.
We are, okay.
Yeah.
There are things called methylation aids, methyl groups on your own DNA.
Don't go there.
I mean, it's yours, okay. John doesn't like his thundersteels, and since he's 6'12", I'm not going to argue with him. So,
Thank you, Frank.
it's clear. So it's modifiable. And there are things that we can do, Doug alluded to them, where we can modify our genetic predispositions.
Thank you. That was terrific. Our next luminary is Dr. Don Lloyd-Jones. Don is the professor and chair of our magnificent Department of Preventive Medicine. He also is in our department as well, in the Department of Medicine, and he's a cardiologist too. That he's going to talk about his work and his perspective on the same questions. Don.
Thank you, Doug. Thank you to all of you as well for being here tonight. And you heard a little bit about where I'm going already. But I'll forgive Frank for that. So we all understand that the genes we inherit, the actual DNA sequence code that we inherit from our parents, has a major sort of impact on our propensity for these chronic diseases of aging we're talking about. Cardiovascular disease, cancer, dementia, chronic kidney disease, diabetes, arthritis. No question that that's locked in at the moment of conception. But what if I told you that you actually have it in your power to change the structure of your DNA, not the code of your DNA, but the structure of your DNA, to actually alter that propensity for chronic diseases of aging, and yes, to extend your longevity as well as your healthy longevity, your health span. Yes, you have this within your power, and that's what I want to talk to you mostly about tonight. So I'm going to leave that there for you to ponder about and talk about the sort of trajectory of research that's been happening in my Department of Preventive Medicine over the last many decades, and then bring it all back kind of home to help you understand how epigenetics plays a critical role in this process of aging. So I'm going to start with telling you the story of the founding chair of my department, Dr. Jeremiah Stamler, who founded the department in 1972. Jerry himself is a story of healthy aging. He passed away last year at the age of 102 and a half. Jerry. 3 days before he died, he was finishing a manuscript and rewriting his R01 grant renewal. Now, if you don't know what an R01 grant is, that is the sort of coin of the realm of NIH funding. If you have an R01 grant, you have arrived as a scientist, basically, in this country. He had had R01 grants, and he was renewing his R01 grant that had been going on for decades, studying many of the concepts we're talking about tonight. He was really one of the founding fathers of cardiovascular epidemiology and prevention. He was among the group of luminaries who defined those things we now think of as traditional cardiovascular risk factors: cholesterol levels, blood pressure, diabetes, smoking, age. And so, he was one of those brilliant individuals who had a full career and then said, wait a minute, we understand risk. Can we flip this paradigm on its head and understand health? And I was very fortunate to come along early in my career, just at the time when he was flipping the script and saying, okay, we understand risk. We've even now got some tools to treat risk, medications, other things we can do to reduce risk for cardiovascular disease in particular. But he said, let's look at the people who aren't developing these risk factors as they age. What's special about them? What do their outcomes look like? And sure enough, he showed us that if you maintain the optimal profile of these cardiovascular risk factors into middle age, you win the game. It kind of is the fountain of youth, right? Not only do you live substantially longer, but you live substantially healthily longer. And he started to show that in some early studies, and that's where then the next generation of folks in our department sort of picked up the ball, and through the early 2000s, really started to define a little bit more about this construct. And we got a real shot in the arm in 2010 when the American Heart Association picked this up and said, this is something important. Let's define this construct as a new thing called cardiovascular health. And so they actually put their quarter down and said, this is what cardiovascular health looks like in individuals and the population. This optimal profile of, at that time, 7 things: a healthy diet, participating in physical activity, avoiding nicotine exposure, having a healthy weight, healthy cholesterol levels, healthy blood pressure levels, and healthy blood sugar levels. So that's what was the original Life Simple 7. And over the last decade, our department has really been digging in on understanding not only what are the outcomes of people who maintain this optimal cardiovascular health profile, but even more importantly, what upstream determinants of cardiovascular health can we manipulate in order to preserve cardiovascular health later into the lifespan? Not only what determinants are there, social determinants, health behaviors, other things that help us preserve our cardiovascular health, but also what are the molecular mechanisms that indicate people who are maintaining better cardiovascular health, things that we could maybe manipulate in order to prolong that health span, prolong that lifespan, and help you avoid cardiovascular health. Well, that construct of cardiovascular health, initially Life Simple 7, was actually updated just last year to Life's Essential 8. Does anybody know what the 8th metric is? Say it loud. Sleep. Sleep, right? We get it, right? All of us get it. You are better when you've had a good night's sleep than when you haven't. And of course, it affects your cardiovascular system in so many important ways. And so what we've shown over the last decade in our department, and now catalyzed by the new institute advancing this research, is just how centrally important this construct of cardiovascular health can be. Notice everything that I mentioned is within your control, largely through healthy lifestyle, right? Largely through a healthy eating pattern, avoiding nicotine exposure, getting and training yourself to get healthy sleep, participating in physical activity. Then those downstream things work better. You have a healthier weight. You have healthier blood pressure levels. You avoid diabetes. You have healthier cholesterol. So this has really come to fruition, I think, in a number of important ways. But I'll give you just one piece of data to let you think about this. So using our construct of cardiovascular health, we've actually looked at people who have a genetic makeup that gives them significantly high risk for cardiovascular disease. So again, that's baked into their DNA code. The whole conglomeration of genes that they inherited from their parents says this person's on the fast track for cardiovascular disease. And we can also see people who inherited a particularly beneficial group of genes that says they should be at really low risk for cardiovascular disease. But what does their cardiovascular health do to modify that? Well, what we see is if we take a group of people with bad genes and good cardiovascular health, and we compare them to people with good genes but bad cardiovascular health, what's the difference in their longevity and their health span? I think it's going to shock you. People with bad genes but good lifestyle, on average, will live 11 years longer than people with good genes but bad lifestyle. And they will live on average 18 years longer without cardiovascular disease during their lifespan than the people with good genes but bad lifestyle. So it really tells you these are modifiable things in our control that can trump those bad genes. How does that happen at the molecular level? It happens largely through epigenetics. So this isn't the DNA code, this is the structure of your DNA. As you go through life and you eat different things and you encounter different things that you inhale or toxins that you drink or eat, it changes your DNA by putting these little groups, these methyl groups, little carbon groups at different points on your DNA. That happens to a certain extent at a predictable rate. So we do have an epigenetic clock, which is a way to tell your biological age. But it also happens in an accelerated way if you are exposed to more toxins, worse diet, less physical activity, more smoking, worse air pollution, that can accelerate your epigenetic clock. And when those little marks get put on your DNA, if they're in the right place, they can switch off your ability to use that gene. Or they can make it so that you can't switch off that gene. So it doesn't matter what the DNA code is anymore. If you can't use it, or if you can't alter when you're going to use that gene, it doesn't matter anymore. The epigenetics actually show the regulatory control of how you use your genome. And that turns out to be functionally a lot more important in many ways for these aging processes. So what we are developing now is we have data on thousands of individuals over time, at multiple time points in their life, where we've done a full epigenetic profile on their entire genome. And we can see what behaviors and environmental exposures influence their epigenetic clock and their epigenetic marks on their genome. And then we can see which one of those are particularly important in driving their chronic diseases of aging. So it's a really important way, and this is now being played out in the Human Longevity Lab, as you heard, as one of the ways that we're going to look at molecular aging and develop, hopefully, a commercial test to tell someone, where are you today? Why do we think you're there today? And if we do this, can we expect that your molecular aging, your epigenetic clock, will actually get better? We can hopefully turn you more into a super ager rather than an accelerated ager. So many threads kind of coming together from traditional epidemiology to now molecular epidemiology and coming to bear on our understanding of aging. So I'll finish there, and to answer your question, Doug,
Yes.
is it inevitable or is it malleable? Yes, it is.
Okay. Both. Our next luminary, maybe our brightest luminary of the evening.
Wow,
Save the best for last, right?
a
Absolutely.
lot of pressure here.
Dr. Farzaneh Sorond, professor of neurology, the young professor of neurology, and also our vice dean for faculty affairs at Feinberg. And she's going to talk about maybe the part of the body and the system that you worry about the most as you age, your brain. So Farzaneh, it's up to you to close this out.
Thank you. Well, I'm delighted to be here, and thank you for joining us tonight. I'm glad you said it. You know, there's an internal joke between neurologists and all the other subspecialists that ultimately all the other organs serve the brain. So, but I didn't say it, you said it. So I'm glad to be going last so that we can talk about the brain. But I fully realize that it is a body and the whole system that is required, and no one organ can live without the other organs. So as we are learning about aging, we go across organs to sort of apply some of the things that cardiologists have learned to the brain. As you know, the brain is a little bit less accessible than the other organs. So to study it, we tend to sort of rely on more accessible organs to test some of our hypotheses. So I'll tell you how I got here and studied aging, and I think there's another neurologist, at least in the audience, who might appreciate this. But 20 years ago, when I was a resident, we had this habit of looking at patients' MRIs before and sort of looking at the records before we went and saw the patient. And I remember looking at, the medical records and looking at the brain MRI and expecting to see, this maybe cognitively impaired, really low-functioning individual, and I would walk in and be fully surprised at this 70-year-old who was a powerful executive and was just there because they had a little thing that they didn't know what it was, and their brains on MRI would tell a completely different story than their functional status. And I remember going back to my attendings at the time and saying, well, why is that? And we don't know. We really don't know. And so people age differently. And so that's sort of was in the back of my mind as I started sort of understanding cognitive aging and really relying on the cardiology world and the research that's been done in the vascular world. Some of the changes we see very prominently on brain MRIs are really hallmarks of vascular disease. And so I started looking at, how do the vascular changes in the system and the brain affect the brain structure? And why is there such a disconnect between the picture of the brain and the function of the brain? And so that sort of line of inquiry that's been over the past 20 years and collaborating with Don since I moved to Northwestern in 2015 has really led us to define what our area of research is, which is understanding the signatures of resilience and vulnerability in the brain. And that is, what is it that makes a structure that looks not the way we expect it to look as far as a healthy brain structure really function well? So what are those signatures in the brain, in the person's life, in their epigenetic modifications, in their genetic code, but also maybe in how the brain is connected that allows them to be so highly functioning. And then you see someone else with pretty much, what would be an age-appropriate brain structure, which we now know that doesn't exist, really doing poorly and having cognitive impairment. In fact, when you look at the brain MRIs of patients with Alzheimer's disease, which we know is really a pathological diagnosis, the biggest, most prominent changes are the vascular changes. And so therefore, this has been this continuous discussion about the spectrum of vascular and neurodegenerative disorders. So in our work over the past 10 years or probably 7 years at Northwestern, we have learned that how you put your brain together really has to do with how your brain falls apart, right? So it goes back to what we heard a little bit about. What was your educational status? What was your socioeconomic status? How did you take care? What was your in utero experience as a fetus, right? That it all starts there and sort of starts building the brain and the body that ultimately lives through the decades of injury that you experience, from cardiovascular risk factors, which really we should call vascular risk factors because they are in the brain as well and not limited to the cardiac system. But the vascular changes that.
You get that?
But the vascular changes.
You got nothing without the pump, baby. That's all I can say.
And if your immune system ain't there, you ain't got it.
There you go. So it's all one word. Yeah, so as you live through those vascular injuries, as you live through pollution, as you learn through stress, sleep deprivation, bad diet, you are going to really fall apart based on how your home was built together. Remember the 3 pigs and the houses they built? So that's really how that is. And we're learning that those who had a stronger foundation in building their brains are a lot more resilient. And what does that resilience mean? It means that they are able to compensate, to develop new networks and pathways in their brains to go around the scar or the injury that happened, which is the same scarring and injury actually we see in patients with HIV disease, with HIV and AIDS. So the real question that we want to answer is, what are those mechanisms? What are those fundamental processes that allows you to rebuild, that allows you to go around and compensate in your brain, that people who actually preserved and put their brain together continue to take advantage of, but those who didn't from a lower socioeconomic status, from lower education, bad nutrition, a stressful life, the type A personality that was actually defined in San Francisco, right? How does that, what are those mechanisms, and can we harness it so that we actually reactivate those? We know that a lot of those processes and genes and mechanisms exist. We just have forgotten how to use them. So that's my work. To get to those questions, we are fortunate to be funded by the NIH to study the signatures of resilience and vulnerability in the brain, and collecting individuals, about 3,000. We sort of go big at Northwestern. 3,000 participants who have those lesions that we now know are hallmarks of vascular aging in the brain, and looking at what brought them there, to understand how they got there, and how are they functioning, to understand what are those single processes, individual processes that then we could sort of study at the molecular level. We're collecting a lot of biomarkers from them, brain imaging, specimens, and really going back in partners from experimental models to understand those mechanisms. And it may be Pi1, who knows? But really thinking about targeting and really reinvigorating those fundamental processes that we all had but lost along the way. And yes.
Yes?
Yes.
Okay. Thank all of you. Those are fantastic discussions, and they're all. It's exciting to hear all of you talk about it. It certainly creates a lot of questions in my mind. Maybe somebody in the audience has a question or two. It looks like a few hands.
Yeah.
Okay. My question was. My question was triggered when you were talking about how people who are being treated for AIDS and it's suppressed age faster.
Yes.
And I remember reading something about survivors of childhood cancer who also age
That's
faster.
right.
And can one tell at this point whether it's the underlying disease or the drug treatment that allowed them to survive so far that has caused the accelerated aging?
That's excellent. I can't speak to childhood cancer because that's not my area of expertise. But it's clear that there is an insult, damage done with HIV, that the later, the more advanced the illness of HIV, at the time that life-saving therapy is begun, the more damage that exists and is not fully recoverable from. It's also clear, though, that even among people who start therapy within days to weeks of becoming infected, which is not the norm, still have the deck a bit stacked against them in terms of the fact that they have an accelerated process of the aging of their blood vessels, the aging of their brain, their kidneys, their lungs, their bones, because of this inflammation and adverse immune activation that takes place. My esteemed colleague at the end here, I want to repeat something because what we call proportional attributability, what to blame for what aspect, is not completely sorted out. In the case of HIV, we have a preponderance of what we call adverse social determinants of health: poverty, mental illness, drug use, obesity, hypertension, diabetes, elevated cholesterol, all the things that we know hasten that biological clock and make it tick faster. So it's a very complex answer to your question, but it's a bit of both.
But fair to say that the early generation AIDS treatments were also not particularly favorable for some of these conditions as well.
A few of them, but when we really look critically at the data, the magnitude of the blame that we can assign to the use of those drugs is minimal compared to what the virus does to the blood vessels and to the immune system.
I'm trained as a mathematician, among other things, and your graph of the superagers, normal, and the accelerated agers is a two-dimensional graph. It wasn't really obvious exactly, but when you were an accelerated ager, you died earlier. Normal, you die a little later, and if you're a superager, you die later still. But that's only two dimensions. So, for instance, let's say you have, what's your mental capacity? That's another dimension. So you could live a very long life, but not even know you're alive, literally. And so I think that aging isn't just two dimensions; it's multi-dimensions. And you know, if you add them all together, even within memory, even within mind, you can have short-term memory loss, still have long-term memory, and so it's really very multidimensional. And of course, you can have you know, mobility problems, right? You might, you know, like Stephen Hawking's a perfect example, right? So it's aging is like a very multidimensional thing, and there's even a philosophical or ethical aspect to it. That how do you want to age? You know, do you want to have a brilliant mind to the day you drop, or do you want to just sort of not know you're going when you're going? You know, so there's a lot of things here, and I wish there was maybe a metric that you could.
So there is a metric of this.
Okay, good.
Actually.
I'm glad I asked you.
So the way we quantify this in my world of epidemiology and you know, thinking about populations is it's a metric called compression of morbidity. Now, the unspoken thing we have not yet said here tonight is, and I'm sorry to have to break this to you, we are all going to die. Show of hands, anybody surprised by that? Okay, good. We are all going to die. But how we get there can, as you've heard, can differ radically, right? And so when we're born, we're actually born with basically the highest stock of health we're ever going to have. And it doesn't mean it's full, right? Some people born in adverse circumstances have a much less full bucket than people who are born in more favorable circumstances. But it's about as full as it's ever going to be. And the normative, to put you on a graph since you're a mathematician, the normative process of aging looks like this until we die. But what we're trying to do, I think, is this, right? We're trying to square that curve so that we are as healthy as possible for as long as possible, and we get sick as close as possible to the date of death, right? That's winning the game, I think, right? So compressing that morbidity as close as possible to the end of the lifespan, that's a quantifiable thing, and especially if you then say, okay, we're gonna discount for heart attacks and strokes that may occur, for dementia as it comes on, for other things, mobility loss, arthritis, things that cause pain. You can actually quantify this and see where someone is in their ability to square that curve, compress their morbidity. And that's one of the metrics that we actually use, you know, compared to normative populations to try to see where somebody is falling.
And I really like your comment. I think that's what the Human Longevity Laboratory is all about. We're collecting information in every accessible dimension right now. And I don't necessarily think that every measure in a given person is going to be synchronized.
Right.
You know, I think there'll be some that'll be faster, some slower, some overall might be different. But yeah, next time we'll bring a four-dimensional hologram to illustrate that and make you happy.
And one other, before we get to the next, because I just want to elaborate a little bit on something Dr. Farzaneh said, is add on to this that there's this whole idea of resilience, that once damage is done, the ability in the case of the brain and in the case of the vascular system and in the case of the immune system to circumvent the injury and to recover and even do better after that injury. We don't, and this concept of resilience is something that we're just beginning to scratch the surface of, and the ability to not only recover, but to augment functional status. And I think we could spend the whole evening just talking about that, but we want to take other people's...
Let's not, though.
Yeah.
Oh, there are more hands than ever.
I'm Grace. I'm 97. I work at Google X, home of the 94 billion parameter model or whatever we're up to now. So this motivates my question. Qualitatively, when you guys talk about the models that you're getting for aging and these causal effects, qualitatively, where are you getting this data? Is it experimental studies or clinical trials? Is it aggregating? And then quantitatively, like how much data are you working with when you talk about this? Orders of magnitude, however you measure it.
We'll talk about that.
Sure. So one of the places that we get our data from is we've actually been part of population-based cohort studies that have followed thousands and thousands of individuals for decades, literally. So one of our studies, the Cardia study, enrolled people at age 18, and they are now passing age 65. This study's been going on for a long period of time. We've seen them over the course of this study now 11 times, poked, prodded, measured, stolen a lot of blood, done a lot of imaging to understand how they've developed risk, how they've lost health, and what sort of preclinical indicators there are that they're aging faster or developing disease, and then we follow them as they've transitioned into clinical things. So those are really powerful tools because that's what's allowed us to then get to very high dimensional data because we have all their DNA. We actually now have all their proteome, all the proteins that are circulating in their blood. We have their metabolome, all the little molecules that are results of your diet and all the things that your tissues and cells are doing every day. We have their microbiome, the bacteria in their gut. We have their transcriptome, RNA that their cells are actively transcribing. And we have their epigenome. So we have the full ohm. And we're starting.
Showing off.
Yes, we are. And now we're starting to build out the exposome, right? What we know actually from their zip codes, what their exposure has been to different environmental toxins. So all the ohms kind of come together, and when we've phenotyped these individuals so well for so long, we can learn a ton about the aging process.
In the Human Longevity Laboratory, this will be new data, entered in a multidimensional database, a cross-sectional database. Like I said, I hope to make it global. But we're collecting information in a variety of different domains, including all the molecular profiling that Don just talked about. But we also include things like AI-based interpretations of information. We're using a thing called ECG age, the standard 12-lead electrocardiogram. You run it through the machine learning algorithm. It can tell you if you're going to have an event in the next 7 years. In a way, and I'm a cardiologist, Don's a cardiologist, it can see things I can't see. And we're applying AI to a number of different dimensions of measures that we're using, and I hope that this database will be a one of a kind around the world eventually to understand human aging.
If I may just add one thing, it's clear, and I've spent most of my 30-plus career studying longitudinal databases among people with versus comparable people without HIV. In fact, the longest known history is called the Multicenter AIDS Cohort Study, which is established by the National Institutes of Health in the early 80s, and we are still following these men and women over, some of them over 40 years now, to study, to characterize them, sample their blood, sample their urine, sample other bodily, characterize their clinical events, their drug exposures, their geocoding, what zip codes they lived in, social determinants of health, all the things that we've mentioned. And we've been able to tease out, and as Doug alludes to, the more data points we have on an individual regarding who they are genetically, metabolomically, proteomically, glycomically, the better we are able to decipher what it is that they were born with versus what exposures it is that help determine and modify the risks that they were born with or without, and where the points are that we can intervene. Meaning to say, what are some potential targets that we can decipher from this massive data we have about people that could be the place where a therapeutic intervention, be it a drug, be it a vaccine, be it a lifestyle change, be it some other intervention that can make a difference.
And you sort of, just to add to that, obviously you forgot the exosome.
I did forget that.
So just to
My
add to
bad.
that. So I work with Don on the cardiac cohort and have been looking at the brain of these individuals. So that's sort of part of the population-based epidemiological data that we've been working together on. But I also partner with experimental labs where they look at, we don't have primate models for the diseases we're studying, and we can go into why that is. But we do have rat and mouse models that we sort of go back and test some of these findings and partner with them and we publish together on sort of making sure that there is that sort of bedside to bench and back to bedside concept that's really at the core of everything that we do.
Steve Kirshner, 78, Biomedical Engineering Tech Institute. I have two questions real quick for the first and second speaker, respectively. First question for the first speaker is, you mentioned anti-inflammatory, inflammation as being a cause of aging, and anti-inflammatory drugs, to my knowledge, most anti-inflammatory drugs tend to have a proviso that they cause kidney disease, like Advil and things like that. So just curious about if there's any anti-inflammatory drugs that don't give you another factor for dying early of something else. And the second question, I'll ask it now so that I can give the mic away, is with respect to the methylated DNA. My understanding is that the DNA is in every cell, and the cells are replicating all over the body. If you get some methylated DNA, is it in one cell? Is it in one system? Is it in one organ? Where are these methyl groups going? How can we get? Where do we get them? Hopefully, don't get them from Ethel because we had some wine outside.
You better get through your answer quick.
If you get methylated, how do you get unmethylated?
Here.
Thank you. Okay.
So excellent questions, superb questions. There are all different types of anti-inflammatories. You're speaking specifically what we call NSAIDs, non-steroidal anti-inflammatory drugs. There are many other. Yes, and a potential adverse impact there is upon the kidney. When we more broadly talk about anti-inflammatories, they can include everything from glucocorticoids, steroids. They can include chemotherapy-ish agents like methotrexate. They can include biologics like the newer fangled drugs that we use to treat rheumatoid arthritis, lupus, sarcoid, inflammatory bowel disease. They can include other types of drugs that I'm not going to go into. So there's many, and they can include, and I'm not going to say, even statin drugs. Cholesterol-lowering agents have modest but significant inflammation-lowering effects, including in addition to lowering cholesterol. So there's many ways to skin that cat, and many have been studied with interesting results regarding the potential benefit. One of them is about to be published, and I was involved with it, and Don is on the Data Safety Monitoring Board, which demonstrated, and if I can just take 15 seconds, among 7,000 people, over 7,000 people with HIV who did not meet national cardiovascular criteria for use of a statin, for use of cholesterol, because their cholesterol was too good, that half of them were given a cholesterol-lowering drug called pitavastatin. Half of them weren't. After 7 years, it became clear that these people, all of whom started out cardiovascularly healthy, that by the end of the 7 years, a third of cardiovascular-related deaths and what we call significant disease endpoints were reduced by just giving that statin to people who we wouldn't thought needed it. So that's my shortest answer I can give you.
Wonderful. So one of the great things about the epigenome is it's dynamic. So to answer your question, yes, you can demethylate a site. So there are parts of the genome, we're still really trying to understand this. So there are parts of the genome where if you get a methyl group, it sticks and you're done. And there are parts of the genome where it is quite a bit more dynamic. There's sort of hotspots that can get a methyl group, get it off. And again, that seems to be related to intensity of exposures or healthiness of lifestyle or diet or whatever that is. We're trying to parse some of that out. But the dynamic nature of it is what's really exciting, is that we can use it as a clock because these things can turn on and off. And you can actually change your epigenetic age through some of these interventions that we've talked about. What's really fascinating is, yes, when you are conceived, you inherit an epigenetic pattern from mom and dad. And when you're young, it's much more plastic across the entire genome. As we age, more and more of those methyl groups tend to get locked in and stuck. But especially when we're young, we do inherit this epigenetic pattern, especially from mom. And that's perhaps why the intrauterine environment we mentioned is so important, because it does tend to start to program how we're going to use our genes. So we know that moms who are exposed to either too few calories or too many calories during pregnancy have babies, both of whom are more likely to struggle with obesity. And that's epigenetic programming that's happening in utero to certain genes because the methyl groups are going to differ. But again, that can be interrupted and changed because of the dynamic nature of this. So epigenetics seem to be one of the ways that we intergenerationally transfer propensity, not just through the DNA code, but also propensity for these chronic diseases of aging as well.
There is a universal component to this epigenetic measurement. Steve Horvath and his group at UCLA have studied over 100 mammalian species, everything from mice to whales, and the epigenome predicts lifespan.
Right.
Very, very accurately. And you can take samples from skin or blood or whatever you want, and it tends to register pretty similarly in a given individual across any tissue where it's collected. So it's pretty remarkable. Horvath has this algorithm that's probably the most popular out there today, probably the most accurate, he calls it grim age, kind of tongue-in-cheek. Multiple components, but only looks at DNA methylation sites over a few hundred sites in your whole genome. But it can pretty much tell you what day you're going to die and what you're going to die of if you want to know. Pretty amazing. And it only gets better. And iterations of this are coming forward all the time, and it'll only get better. But it's pretty powerful.
That's a crystal ball not all of us want to know.
Stuff, yeah. Yeah.
Peggy Prendergast, Medill, 94. Part of the question was already answered in terms of database. So Google has some subsidiaries, Project Baseline and Verity, Genentech, another company. I'm sure you're leveraging maybe a lot of that database already to also increase for the information that's already in the longevity lab. So are you leveraging outside sources and third-party information? And the second is, what's the net new? What can we as attendees take away from all of this? And what can we go home with and learn and understand our own longevity and how we can ameliorate that?
Great question.
Great.
Doug, you should do that.
Well, I think that Don is one that leverages outside databases more than any of the rest of us, and I think his department does that in a variety of different ways with existing databases. I don't think there's any existing database in the world that is necessarily as multidimensional as what we're going to collect at Northwestern. I'm pretty confident of that. No database has been developed with the insights from a chair of medicine that thinks about every organ system. And so we'll have that, and they'll give us the opportunity in real time to do interventional trials and to see if we can start to change the pace of aging in individuals that participate in our cross-sectional studies. And so we'll do it in people. We'll do studies in people with chronic kidney disease. We'll do studies in people with HIV. We'll do a variety of other studies in other people. And I don't even know what intervention is likely to be the most interesting. It might be a dietary restriction component. It might be exercise. It might be metformin. It might be some other new class of drugs that comes along. But I think the idea that we're creating a data set that's so robust and so complete, I think that gives us something unique. And what you can take away from tonight, I think, is the reality that you need to stay tuned, that things are happening in this space very rapidly, very quickly. There are going to be ways that you can address your own health and your own health span, I think, in real time that will impact on your life. But I can't tell you exactly what that is today.